Okay, so moving on through this lecture, it moves on to intragenic mapping and how to use bacteriophage infection to look for the order of genes along viral chromosomes. The reality is that's not one of our course outcomes. So we're going to stop right here at this point in chapter 10, because when we start rolling into the next set of chapters, we're going to be moving on to molecular biology. Looking at viral infection in terms of how the infection occurs, okay? How the initial genomic information enters the host cell, how the infection either becomes or how the DNA becomes incorporated into the host genome, or the cell goes into the lytic cycle because of directions that are being expressed from the virus. And then the outcome of all that. That's really kind of what's important. Understanding that you can use viral infection as a mapping tool. I think that we're going to just stop here at this point in this chapter, because that's not really a part of introductory genetics. That starts being a part of molecular biology, microbiology, and upper division genetics. So anyways, lucky you guys. We will be starting chapter 11 next week. The chapter 11 lectures should be coming up pretty soon. I have to make them. And anyways, if you have any questions, send me an email. Send me, start a discussion. There are always discussions. Remember, there's a discussion to participate in on the 3 forms of genetic change in bacterial cells. And I will hopefully see you guys soon. Have a good one.